Hello everyone, welcome to PG Pathshala. In this video, I'll be covering module 12 of Cellular and Molecular Biophysics. This module is on molecular mechanisms of cell differentiation and cell fates. I am Ashik Sethi and currently I work at the All India Institute of Medical Sciences, New Delhi. Let us see the objectives of this module. At the end of this module, you should be able to list various molecular mechanisms of cell differentiation, explain cell fate determination, explain transcriptional regulation of protein expression, explain epigenetic regulation of cell differentiation, and cite examples of reversible cellular differentiation. Cell differentiation is a process in which a cell changes from one form to another. Differentiation usually provides the cell with new functional capabilities. How are these novel functions bestowed upon the differentiated cells compared to the parent cells? This is because of the expression of a new set of proteins in the daughter cell which were either absent or minimal in its predecessor. The cellular differentiation is associated with extensive reprogramming at the transcriptional, post-transcriptional and translational levels, enabling expression of different set of proteins at different levels in the differentiated cell compared to the parent cell. In this module, various molecular mechanisms that are involved in cellular differentiation will be covered, along with the implications of alterations in such mechanisms. Let us first discuss cell fate determination. The process in which a cell irreversibly commits to a particular cell fate is known as cell determination or cell fate determination. How is it that two daughter cells arising from a single parent cell can have different fates? Multiple mechanisms have been postulated to explain this. Some of these are asymmetrical distribution of molecules, inductive signaling, and pattern formation. Cell division can result in asymmetrical distribution of molecules among the daughter cells. For instance, the mRNA content of the daughter cells can considerably vary from one another. If these mRNAs suppose encode transcription factors, this can lead to altered transcriptional regulation and thus altered protein levels in the daughter cells. In certain other cases, it can be the transcription factor themselves that are differentially distributed. The second mechanism of cell fate determination is inductive signaling. Inductive signaling refers to the short-range signaling between cells, where signaling from one set of cells of a different ancestry affects the neighboring cells and causes them to adapt a particular fate. Here, the inducer cells produce a molecule, which is usually a protein, that interacts with the responder cells and leads to alteration in properties, for example mitotic rate, cell shape, etc. The interaction between the inducer and the responder can be mediated by different mechanisms like diffusible signals or direct cell-to-cell interaction or through gap junctions. Inductive signaling is particularly important during organ formation. Pattern formation, the third mechanism, deals with the positional orientation of a cell within an organ. A diffusible chemical known as morphogen can be released from certain cells, and this usually results in the formation of a concentration gradient. Thus, different cells along the vicinity of this morphogen gets exposed to the different concentration of the morphogen, and this leads to differential programming of the cells. It will be interesting to note that the term morphogen was actually coined by the English computational scientist Alan Turing, whose life was depicted in the movie Imitation Game. This image summarizes the mechanisms of cell fate determination. You can see the three mechanisms that I just mentioned, that is inductive signaling, asymmetrical distribution of molecules during cell division, and the pattern formation that involves morphogen, for example like growth factors in this picture. Whatever be the mechanism, cell fate determination is followed by cellular differentiation, and this is associated with an alteration in the protein types and amounts in the cells. Various mechanisms exist to regulate the expression of proteins in a cell. The processes important in cellular differentiation include, but are not restricted to, transcriptional regulation, translational control, gene amplification, and gene rearrangement. Discussion on transcriptional control merits a separate section. Let us now see the other regulatory mechanisms first. Translational control. P-bodies or processing bodies are foci present in the cytoplasm of eukaryotic cells that are associated with mRNA turnover. Not all the mRNAs that enter the P-bodies are degraded. mRNAs can exit from the P-bodies and can further undergo translation. Thus, certain mRNAs that are stored within the P-bodies are temporarily inaccessible to the translational machinery. This means that such proteins can be temporarily expressed depending on the cells involved. Gene amplification. Protein levels can be increased due to the increase in the number of gene copies encoding the protein by selective amplification of the gene involved. Gene amplification is also seen in certain tumor tissues and has been postulated as one of the mechanisms for chemoresistance. For example, MDR or multi-drug resistant protein is involved in the extrusion of certain drugs from the cells. Amplification of MDR gene results in increased MDR protein levels in the cell, and this promotes resistance against chemotherapeutic agents like methotrexate, which are actively extruded from the tumor cells. Gene rearrangement. Gene rearrangement is observed in B and T lymphocytes. The gene rearrangement in the B cell receptor and T cell receptor gene complexes, respectively, in these cells lead to the formation of B and T cell receptors. The specific patterns of rearrangement leads to diversity in the B and T cell receptors, thus enabling the immune system to respond to a wide variety of antigens. Defects in B and T cell receptor rearrangements can lead to immune deficiencies. This image shows the gene rearrangement in a B lymphocyte. You can see the immunoglobulin heavy chain gene locus, which is comprised of multiple segments, that is V, D, J and C. The VDJ region forms a variable region while C forms a constant region, and as you can see, the variable region is composed of V, D and J, which are again of multiple segments. One V, one D and one J recombine and they form a single unit which combines with the heavy chain to form the mature immunoglobulin heavy chain. Let us now discuss transcriptional control in detail. Expression of different sets of proteins is mainly regulated at the transcriptional level. Transcription factors can bind to the DNA at the promoter and enhancer regions and can promote the recruitment of RNA polymerases. This results in increased transcription, thus increased mRNA levels and increased protein levels. Repressors are molecules that antagonize the action of inducers. They can bind to promoters or to DNA regions known as silencers. Transcriptional control is achieved mostly via epigenetic mechanisms. The cell differentiation is an epigenetic process. Epigenetics refers to the heritable changes in the genome without any change in the sequence of DNA. Epigenetic mechanisms play a prime role in the regulation of gene expression. DNA methylation and histone modifications are the major epigenetic mechanisms. 5-methylcytosine, that is 5-MC, is an epigenetic modification which exists in the CpG dinucleotide context within the DNA. 5-methylcytosine is also known as the fifth base of DNA. These are commonly found in the 5' end of the genes. The methyl group of the 5-methylcytosine protrudes into the major groove of the DNA where it interferes with the binding of transcription factors. CpG methylation is mediated by a group of enzymes known as DNA methyltransferases, and the removal of this signature, demethylation, is mediated by enzymes like 10-11 translocases. Now 10-11 translocases convert 5-methylcytosine to 5-hydroxymethylcytosine, which is further removed by DNA repair mechanisms. 5-hydroxymethylcytosine is also known as the 6th base of DNA. Eukaryotic DNA is restricted to the axis of transcription factors. This is because of the compaction and packing of the DNA, which is mediated by the histone proteins. In this image, you can see the action of histone acetyltransferases on histones. So just as I mentioned, histone acetyltransferases, they add an acetyl group with the help of acetyl-CoA to the lysine residues of histones, and this results in the loss of the positive charge, thus decreasing the interaction between DNA and histones, leading on to unwinding of the histones. And this unwinding makes the histone more accessible to various proteins, including transcription factors. The other major histone modification is the methylation of amino acid residues of histones. Lysine and less commonly arginine are the amino acid residues that are subjected to methylation. Unlike acetylation of lysine residues, histone methylation can increase or decrease the transcription based on the position of the residue that is methylated, as well as the number of methyl groups that are added. Lysine residues can be mono, di or trimethylated, while arginine residues can be mono or dimethylated. Addition of methyl groups to lysine is catalyzed by histone methyltransferases, while the similar reaction involving arginine is characterized by protein arginine methyltransferases, also known as PRMTs. S-adenosyl methionine, also depicted as SAM or Adomet, acts as a methyl donor in these reactions. In this image, you can see the histone methylations. Arginine residues are methylated by PRMT, that is protein arginine methyltransferases. Remember, R is the single letter short form for arginine, that is why the enzyme is named as PRMT. So arginine is first monomethylated, and another PRMT acts on this monomethylated arginine residue to form a symmetric or symmetric dimethyl arginine. Addition of methyl groups to lysine is catalyzed by histone methyltransferases to form mono, di or trimethyl lysines. How exactly does histone methylations alter transcription? There are multiple mechanisms involved, but the prominently identified mechanism is that the methylated amino acid residues can alter protein binding to histones. Thus, methylation can control the recruitment of transcription factors to histones. The other class of proteins that are differentially recruited in response to methylation is the histone deacetylases. Through all such mechanisms, the rate of transcription can be altered as discussed previously. Let us now see the clinical significance of epigenetic mechanisms in cellular differentiation. Aberrations in the epigenetic mechanisms are associated with arrest of differentiation. This, if happens in hematopoietic stem cells, can lead to abnormality in the formation of mature blood cells like RBCs, WBCs and platelets. This can lead to anemia, leukopenia and infections, and thrombocytopenia and bleeding. This is what exactly happens in myelodysplastic syndromes, which is associated with mutations in 10-11 translocases or DNA methyltransferases. So, hypomethylating agents such as decitabine are used in the treatment of such conditions. HDAC, that is histone deacetylase inhibitors like SAHA, are already in use in the treatment of conditions like cutaneous T-cell lymphoma. Let us now see some more examples for the role of proteins in cellular differentiation. I'll briefly discuss 4 topics: somatic cell nuclear transfer, induced pluripotency, direct reprogramming, and the Hedgehog pathway. I'll briefly discuss 4 topics: somatic cell nuclear transfer, induced pluripotency, direct reprogramming, and the Hedgehog pathway. Somatic cell nuclear transfer. In somatic cell nuclear transfer, the nucleus of a somatic cell is transferred to the cytoplasm of an enucleated cell, usually an ovum. Various factors present in the cytoplasm now leads to reprogramming of the somatic cell nucleus, conferring pluripotency, leading to the generation of embryonic stem cells. These cells can reach the blastocyst stage when they can be used to isolate embryonic stem cells, or the blastocyst as such can be introduced into the uterus of a surrogate mother, which is what is done in the case of reproductive cloning. You might be familiar with what Ian Wilmut did for reproductive cloning by generating Dolly. Induced pluripotency has been described in detail in module number 8. Briefly, the introduction of selected transcription factors like SOX2, OCT4, KLF4 and c-MYC is sufficient enough to induce pluripotency in differentiated cells. Studies have shown that these transcription factors can lead to alteration in DNA methylation patterns and histone modifications, thus enabling the expression of genes that regulate pluripotency. Direct reprogramming. This refers to the ability of an adult cell type to become another adult cell type without passing through a pluripotent state. The classical example of direct reprogramming is the conversion of fibroblasts to skeletal muscle cells by ectopic expression of a single gene, that is MyoD1 or myogenic differentiation 1 gene. All these examples of reversible differentiation or reprogramming that I just mentioned accentuate the fact that differential protein expression at different phases of cell development leads to cellular differentiation. Let us conclude by explaining the role of signaling pathways in cellular differentiation. Though various cellular pathways like Wnt beta-catenin pathway and Notch pathway are involved in cellular differentiation, I'll restrict this discussion to the Hedgehog pathway. In 1950s, farmers in Idaho, United States, observed that lambs born from eves that grazed in the alpine region suffered from severe craniofacial deformities. Since the common deformity in the lambs was the presence of only a single eye, they were called cyclopic sheep and the condition was called cyclopia. In this image, you can see a one-eyed lamb. Since one-eyed monsters in Greek and Roman mythology were called cyclops, these lambs were called the cyclopic sheep. The reason for such deformities in this cyclopic sheep remained an enigma, and the Department of Agriculture was requested for investigating the probable cause. Extensive studies were conducted to determine the mechanism behind the abnormalities, but all these were in vain. The sheep which grazed on the plant corn lily, that is Veratrum californicum, suffered the most, and this turned out to be the key information in identifying the culprit. In 1960s, the toxin responsible for the birth defects was isolated from Veratrum californicum, and this was named cyclopamine. Further studies confirmed that cyclopamine was responsible for the teratogenic effects in the lambs and restricting sheep access to Veratrum californicum during the gestation, thwarted further events of craniofacial deformities in lambs to a significant level. In 1970s, two scientists, Eric Weishaupt and Christian Nusslein-Vollard, were studying the embryonic development in Drosophila melanogaster, and they observed that inactivating a particular gene resulted in the formation of flies with spiny projections resembling a hedgehog. Hence, they named the gene hedgehog. Later, mammalian counterparts of the hedgehog gene were identified, and these were named as desert hedgehog, Indian hedgehog, and sonic hedgehog genes. Weishaupt and Nusslein-Vollard were awarded Nobel Prize in Medicine or Physiology in 1995 for their contributions to developmental biology. Hedgehog proteins are responsible for a signaling pathway that has a crucial role in cellular differentiation, especially in embryonic cells. The levels of hedgehog proteins differ in different parts of the embryo. In mammals, including humans, sonic hedgehog gene SHH was found to be responsible for symmetrical body development, and the defect in this gene leads to severe developmental anomalies including cyclopia. Hedgehog proteins have also been found to have roles in maintenance of stem cells, and certain cancers have been found to arise due to the aberrations in the hedgehog signaling pathway. In this image, if you see the extreme right side, you can see the sonic hedgehog signaling pathway. The SHH protein interacts with a transmembrane protein known as PAST. The default role of PAST is to inhibit another protein called smooth-end. Once SHH interacts with PAST, smooth-end is released, and this leads to activation of transcription that is required for growth and differentiation. Since the presentations of cyclopamine toxicity and sonic hedgehog inactivation lead to similar symptoms, scientists explored the possibility of inhibiting sonic hedgehog mediated signaling pathway using cyclopamine, and they were successful, as you can see in this image. But due to the adverse effects posed by cyclopamine, other inhibitors of sonic hedgehog pathway were developed. At present, 2 such drugs have been approved by US FDA. That is sonidegib and vismodegib, and these are used in the treatment of basal cell carcinoma, which is a malignancy that happens in the skin. For reasons that are too obvious to state, both these drugs are contraindicated in pregnancy. So to conclude the hedgehog story, activation of transcription is a major event in cellular differentiation. Various signals like hedgehog signaling and notch signaling can promote cellular differentiation. Magnitude of these signals varies according to the cell types. Aberrant cellular differentiation can lead to abnormalities in organ formation and function. Aberrant signaling across cellular differentiation pathways can promote tumorigenesis, and inhibition of such pathways have a potential to curb tumor growth. So, to summarize this module, the process in which a cell changes from one form to another is known as cellular differentiation. Differentiation is usually associated with increased functionality and division of labor. The process in which a cell irreversibly commits to a particular cell fate is known as cell determination or cell fate determination. Cell fate determination precedes cellular differentiation. Cell fate determination is mediated by multiple mechanisms like asymmetric distribution of molecules, pattern formation, and inductive signaling. Cell differentiation is regulated at transcriptional and translational levels. Gene amplification and gene rearrangement can also play a role in specific situations. Epigenetic mechanisms can regulate transcriptional control. Methylation of cytosine and histone modifications are the 2 major epigenetic changes. Various signaling pathways like hedgehog signaling pathway can promote cell differentiation. That's all for this module. Thank you.